Good morning everyone. Today I would like to present the introduction for our microbiological analysis experiment using spices powder as the sample. Microbiological analysis is important to determine the quality and safety of food products by identifying the presence and number of microorganisms. Spices powder can easily be contaminated during harvesting, drying, processing, packaging, and storage. Even though spices powder has low moisture content, some microorganisms are still able to survive under suitable conditions. In this experiment, several microbiological tests were conducted using different types of media. Plate count agar or PCA was used for total plate count to determine the total number of viable bacteria. Dichloramose bengal chloramophenicol agar, which is the RBC agar, was used for yeast and mold count, while tryptone glucose extra agar or TGE agar was used for thermophilic bacteria analysis. Serial dilution and pour plate method were carried out to determine the microbial count in the sample. Both aerobic and anaerobic incubation conditions were also used because different microorganisms require different oxygen conditions for growth. Aerobic microorganisms grow in the presence of oxygen, while anaerobic microorganisms can grow without oxygen. Therefore, this experiment helped to evaluate the microbial quality and safety of spices powder based on the microbial loads present in the sample. Thank you.
First, with 10 grams of spice sample. Add 90 ml of saline. Then we put into stoichiometer to homogenize the sample. The homogeneous solution was now known as the first dilution. From the first dilution, we pipette 1 ml to create Adler's dilution. We create anti-acid dilution as shown in the picture. Then we took 12 ml of DCPA that were prepared by lab assistant. Then we label the test tube into aerobic and anaerobic. From dilution 3, 4 and 5, we pipette 1 ml into DCPA test tube. After that, the test tube were heated in 80 Celsius for 30 minutes. Then cool down for 5 minutes before pour the agar into the plate. Then the anaerobic agar were overlaid with diavolactic agar that were conducted by lab assistant and incubated in 37 Celsius for 24 hours. Meanwhile, the anaerobic condition were incubated in 37 Celsius for 48 hours. Then we took the PCA, the RBC and TGE agar that were prepared by the lab assistant. From the dilution for 5 inches of the homogeneous sample, we pipette 1 ml into the plate. Then we put the RBC, PCA and TGE agar, whereby the RBC were left at room temperature for 3 to 5 days, PCA were incubated at 37 Celsius for 24 hours, and TGE were incubated at 55 Celsius for 48 hours.
Today, I will present the results for the microbial quality analysis of ground spices, which is chili powder. First, the total plate count using PCA media. The results show that the dilution factor of 4 was TNTC, while no colonies were observed at 5 and 6 dilutions. This indicates that the chili powder contained a high number of microorganisms at lower dilution levels. Next, for the yeast and mold analysis using the RBC media, only 1 CFU was observed at the 4 dilution while no colonies were detected at higher dilutions. This shows that the yeast and mold contamination in the sample was very low. For TGE, 16 CFU were recorded at 4 dilution. This result suggests the presence of microbial growth in the chili powder sample. Moving on to aerobic bacteria analysis, the highest colony count was observed at 3 dilution with 38 CFU, followed by 5 CFU at 4 and 3 CFU at 5. This indicates that aerobic microorganisms were present in the sample. For anaerobic bacteria, 28 CFU were observed at 3 dilution, 6 CFU at 4 and 28 CFU again at 5 dilution. This shows that anaerobic bacteria were also detected in the chili powder. In conclusion, the chili powder sample contained different types of microorganisms, including aerobic and anaerobic bacteria, while yeast and mold contamination remained low. Overall, proper handling, storage, and hygiene practice are important to maintain the microbial quality and safety of ground spices.
Assalamualaikum and good morning everyone. Today I will be presenting the discussion for our laboratory experiment on the microbiological analysis of chili powder. This discussion focuses on the microbial results obtained from several tests including total plate count, yeast, mold count, TGE result as well as aerobic and anaerobic bacteria analysis. These tests were conducted to determine the microbial quality and safety level of the chili powder sample. For the total plate count result, the plate at 10-4 dilution showed TMTC, which means too numerous to count. This indicates that the chili powder sample contained a very high number of microorganisms. Meanwhile, no colonies were observed at 10-5 and 10-6 dilution, showing that the microbial concentration decreased as the sample became more dilute. The high microbial load may be caused by poor handling, improper processing, and unsuitable storage condition during production and distribution. These results suggest that the sample may have been exposed to contamination before analysis. For the yeast and mold test using the RPC medium, only one CFU was detected at 10-4 dilution, while no colonies were observed at higher dilutions. This suggests that the chili powder sample had low fungal contamination. According to Choice Renewal and colleagues in 2020, most Thai chili powder samples contain fungal contamination below 10.3 CFU per gram. Next, for the TGE result, 16 CFU were recorded at the 10-4 dilution. This indicates the presence of microorganisms that were able to grow on the medium used, showing moderate microbial contamination in the sample. Although the contamination level was not extremely high, it still reflects the importance of maintaining proper hygiene during food processing and storage. Aerobic and anaerobic bacteria. For aerobic bacteria, the highest colony count was observed at 10-3 dilution with 38 CFU. Lower colony counts were seen at the 10-4 and 10-5 dilutions. This confirmed the presence of aerobic microorganisms in the chili powder sample. According to Mendoka and colleagues in 2020, these bacteria are able to grow in oxygen at normal food temperatures. For anaerobic bacteria, 28 CFU were recorded at both the 10-3 and 10-5 dilutions. The presence of anaerobic microorganisms may indicate contamination during processing or storage, especially in environments with limited oxygen exposure. This shows that chili powder can support the survival of different types of microorganisms if not handled properly. In conclusion, the chili powder sample contained various microorganisms, including aerobic and anaerobic bacteria. However, yeast and mold contamination was relatively low compared to bacterial contamination. These findings highlight the importance of proper hygiene practice, suitable packaging, and proper storage conditions to maintain microbial quality and ensure food safety. Proper handling during processing and distribution is also essential to reduce contamination risk in food products.